Hello everybody, this is Dr. Vishal Trivedi from Department of Biosciences and Bioengineering, IIT Guwahati, and in this particular module we were discussing about the different types of the immunological tools which are mainly being based on the interaction between the antigen and the antibodies. So, what we have discussed so far is as follows. We discussed about if the antigen is insoluble in nature, then you can be able to perform the acutylation reactions. If the antigen is insoluble in nature, then you can be able to perform the precipitation reactions or the radial immunodiffusion, and in the previous lecture we have also discussed about the immunoprecipitations. Apart from that, we have also discussed about the different types of immunoassays such as the ELISA, Western blot and so on. So, with these tools you can be able to perform different types of experiments and you can be able to answer many types of immunological questions or the questions which are related to the biology. Apart from that, there is a additional thing which you can also explore with the help of the immunological tools. So, you can imagine that a cellular response of a cell like a immunological cell like macrophages or B cell or T cell. For example, in this cell, in this case it is the T cell which is actually responding to a extracellular ligand, and in response to that it is actually driving a downstream signaling, and it is involving all these signaling molecules, and at the end it is activating the NF kappa B transcription factor. So, NF kappa B is going inside the nucleus and then it is activating its target genes, and in response it is actually producing the different types of proteins. It is actually changing the transcriptional activities within the cell, and as a result it is actually going to make a complete cellular response, whether it is this is going to be a humoral cellular response or to the cellular cell response. Whether it is going to be a humoral immunity or the cell mediated immunity, but at the end what you are going to see is that in the event of any such signaling, what will going to happen is that it is actually going to change the gene expression of that particular cell. It is actually going to change the metabolism of that particular cell, or it is also going to change the proteins of the cell. And if this particular cell is a immune cell, for example the T cells or the B cell, it is also going to change the cytokine secretion from this particular cell. So, if you have these particular 4 parameters which you would like to monitor, you have different types of techniques. For example, if you want to monitor the gene expression profiling, you can do that simply by using the microarray chips. If you want to study the metabolomics or the metabolic changes, then that can be done simply by the with the help of the NMR techniques. And if you are interested to see the changes within the proteins, then you can be able to do with the proteomics. But what would be the technique if you would like to study the complete cytokine profiling of a cell under a particular cellular response? So, to this we have the multiple options. One of the easiest way is that you can perform the ELISAs. So, I think in the previous lecture also we discussed about the different types of ELISA, what are available for you to explore. And see, so the cytokines are actually the signaling molecules. So, cytokines or the chemokines are the signaling molecules that mediate the cell to cell communications. They are released from the cells and have the critical role in many biological processes such as the cell growth, differentiations and gene expressions, migrations, immunity and the inflammations. In most biological processes, the multiple cytokines operate in a large network where the action of one of the cytokine is regulated by the presence or the absence of the other cytokines. This means the cytokines are actually very versatile in nature. So, they can be secreted from one cell, they can go to the 5 more cells, and then that is how they can actually be able to make a very complicated network where the one cytokine is regulating the secretion as well as the action of the multiple cytokines. So, what is the option what we have to measure these many cytokines from a single cell, that too at a particular moment. So, if you remember in our previous lecture we discussed about the different types of immunological tools what you can actually use to measure the antibody or the antigens. So, ELISA is one of the immunological tool that what we have discussed, and we have discussed about the different types of ELISA like the direct ELISA, indirect ELISA, sandwich ELISA as well as the competitive ELISA. So, if you remember that the sandwich ELISA is something what you can use to measure the level of the antigen. So, sandwich ELISA can be a potential immunological tools which actually you can use to measure the level of these cytokines, but the number of cytokines are very big. So, if you perform the sandwich ELISA for the individual cytokines, it is actually going to be a very laborious work, and on the other hand it is actually going to give you it is time consuming, and as well as it is actually going to cost you a lot of money. So, instead of doing a sandwich ELISA, the people have developed the cytokine arrays where you can be able to measure the level of cytokines at a particular moment from a cell. So, whether you use the cells or the cell supernatant, or whether you use the cell lysate, or whether you use the tissue, you can be able to measure the different types of cytokines. So, how the cytokine array works? So, in a typical cytokine array what you have is you have a membrane on which the antibodies are being immobilized. So, these individual antibodies are directed against a particular cytokine, like so one antibody is for a single cytokine, and you have multiple antibodies, and then what you are going to do is once you add the samples, these samples are the different cytokine molecules. So, they will go and bind to their respective antibodies, and once you are done with this, then you can do a washing, and then you are actually going to add the biotinylated antibodies. See, these biotinylated antibodies are also monoclonal antibody, and they will be also against this particular cytokine. So, you are actually going to have the multiple monoclonal antibodies which are biotinylated, and all these multiple antibodies are going to bind to its respective cytokines, and ultimately at this stage again you are going to wash, and then you are actually going to add the stabdavidine, which is actually linked either with a fluorophore or it is linked to an enzyme, and that actually will allow you with to do the reactions to visualize the samples. And what you are going to see onto the array chip, that you are going to see the individual spots glowing up after the reactions. Either they will going to give you the fluorescence signal, or they are going to give you the chemiluminescence signals. In some cases you can also use the X-ray films to acquire the image or acquire the data from these cytokine array chips. And ultimately what you can do is you can just capture the image, and you can be able to do the analysis of this particular signal. So, the question comes how cytokine array is different from the sandwich ELISA. So, in the cytokine array, the carefully selected capture antibodies are being spotted in duplicate onto a nitrocellulose membrane. The sample or the antibody mixture is then incubated with the cytokine array membrane. Any cytokine detection complex present is bound by its cognate immobilized capture antibody onto the membrane. Following a wash to remove the unbound material, stabdavidine, HRP and the chemiluminescence detection reagents are added subsequently. Light is produced at each spot in proportion to the amount of cytokine present. So, this is actually a cytokine array where you have a membrane where the antibodies are being immobilized, and these antibodies will bind the cytokines which are present into your sample. The cytokine array and the complete idea of doing a cytokine array is very much similar to the sandwich ELISA, except that in this case you are using the different antibodies, and as well as you are actually it is just like that you are performing the 40 or 50 ELISA reactions at the same time, but the only issue is that the antibodies are immobilized onto the nitrocellulose membrane and you are using 2 antibodies just like as sandwich ELISA. If you remember, in a sandwich ELISA also we have a capture antibody which actually going to capture the antigen from the reactions, and then you are actually adding the additional monoclonal antibody which is actually going to recognize the antigen, but with not with the same epitope, it is going to recognize with the different epitopes, and then you are actually adding the secondary antibody which is coupled with the enzyme, and then you are adding the substrate, and that actually is going to give you the signal. So, it is exactly the same, that cytokine array is nothing but the sandwich ELISA which is you are performing, but it is present onto the a thin sheet of membrane instead of doing it in the well. Let us see how to perform this. So, to perform the cytokine array, what you need is you need the cytokine array from a particular company. You require the buffers, different types of buffers, then you require the wash buffers, you require a detection antibody. Detection antibody is also going to be the monoclonal antibody directed against a set of cytokines for which the array is also being designed. Then you need a streptavidin HRP complex that actually is going to recognize the detection antibody, and then you require the chemi reagents to detect to generate the signal. So, this you required for generating the signal, and then you require the well or dish where you are going to do the incubations with the cytokine arrays, or you also require a cover so that you will evaporate, you will protect the evaporation and other things. Then how to reconstitute the different types of reagents. So, in the step 1 or what is the procedure in the step 1, you are going to prepare the samples, and you are going to have the multiple types of sample like cell culture, cell lysate, you can have the serum, you can have the plasma, and you can have the tissues, and the processing of all these samples are different, except that you have different depending on the type of the contaminating agents what is present and how to process. For example, in the case of cell culture supernatant, you do not need to do anything except that you remove the particulate matter, and then you are simply can use these supernatant into the cytokine array assay. The only thing what you have to avoid is you have to avoid the repeated freeze-thaw cycles. Similarly, for the cell lysate, you might have to pass through the cell lysate through 0.22 micron filters and other kind of things, so that there will be no interfering agents present. And then you have to do protein estimation, so that you will not going to add lot of cytokines and very little cytokines, otherwise the detections are going to be not perfect. Similarly, the similar kind of thing what you have to do for the serum as well as the plasma, and the tissue analysis is also being done with a set of protocols which you have to follow, and ultimately what you are going to get by following any of these processes is you are going to get a mixture of the proteins and where your cytokines are present, and then the idea is that you should use that particular cell mixture or the protein mixture immediately, or you just keep it on a -20, you avoid the freeze-thaw cycle, and then also you should do the protein estimations, so that you should not see a lot of variability from your results from one batch to another batch. So, the reagent what you need to prepare, the reagent is that you have to prepare the cytokine array. So, you are going to get the nitrocellulose membrane which actually contains the cytokine antibodies. The only thing what you have to do is, because these are very sensitive membranes, so you should not handle the membranes only with the gloved hand, and you use the tweezers rather than the hand or the gloves. So, you use the gloves and as well as you use the tweezers. Then you have to prepare the detection antibody. So, what the company is going to give you, it is going to give you a concentrated stock, and then you have to simply dilute the concentrated stock and reconstitute with the help of the buffers what they will provide, or you can use the distilled water Then you have to prepare the wash buffer, you have to prepare the chemi reagents, and all these you are going to use or you are going to prepare as per the instructions what the company is going to give you in the leaflets. And then how to perform the assays. So, bring all, before you start this process, you have to ensure that all the reagents are on the room temperature, and then you incubate them and your sample is on the ice. To avoid the contamination, you have to wear the gloves while performing the procedures, because you know that the human hands are also contains the different types of, it is made up of skin. So, skin cells are very fragile. So, when you are doing all these procedures, the skin cell also can actually be peeled off, and they can actually also be able to contaminate your sample, and that actually is going to give you the variability. That is why you have to wear the gloves in case, so that you should not add the proteins from your body. First you prepare all the reagent, and as we discussed, then you are actually going to do the you have to wet the buffer, and then you have to use the assay buffer 6 as a block buffer. Then you have to use the flat tip tweezers and remove the each membrane to be used between the protective sheets and place it in a well of 4 well multi dish. The number on the membrane should be facing upward. So, the membrane, when you are, it is been kept in a between the two sheet of polythene, and that has to be removed, and then this membrane has some printed material, and that printed material should be facing upward. Then you incubate this with your sample on a rocking platform and orient the tray so that each membrane rocks end to end in its well. While the membranes are blocking, prepare the sample by adding 1 ml of each sample to 0.5 ml of array buffer 4 in a separate tubes. Adjust to the final volume of 1.5 ml with the array buffer 6 as necessary. Then you add 15 microliter of reconstituted mouse cytokine array detection antibodies cocktail to each prepared sample. Mix and incubate for room temperature for 1 hour. Then you incubate the overnight in a rocking platform shaker. Carefully remove each membrane and place it into the individual plastic container with 20 ml of 1x wash buffer. Rinse the 4 well multi dionyls or dry thoroughly. Wash each membrane with 1x wash buffer for 10 minutes on a rocking platform. Repeat 2 times for a total of 3 washes. Then you are going to do a detection. So, with this you are going to do a detection of the sample. So, now your membrane is ready to be detected by the Stavdavidine HRP system. Then you pipette the 2 ml of diluted HRP mixture into each well of the 4 well multi dish. Then you carefully remove each membrane from its wash container, allow the excess buffer to drain from the membrane, return the membrane to the 4 well multi dish container, the diluted this and that, cover the well with the lid, incubate for 30 minutes so that the Stavdavidine HRP complex will go and bind to the biotin-related antibodies. And then you remove the membrane from each well, allowing the excess wash buffer to drain off by blotting the lower edge onto the paper towel. Place each membrane onto the bottom sheet of the plastic sheets protector with the identified number facing up. Then pipette 1 ml of the chemi reagents onto each membrane, and then carefully cover with the top sheets of the plastic paper protector. Gently smooth out any air bubble and ensure that you are going to spread evenly to all corner of each membrane. Incubate for 1 minute, and then you are going to acquire the image, because once you add the chemiluminescent reagents, you have to mate for the 1 minute, so that the substrate what is present in the chemi reagents are going to be processed by the enzyme, and then it is going to give you the signal, and then you have to capture the image. So, then you are going to wrap the excess plastic membrane around the back of the sheet protector, so that the membrane and the sheet protectors are completely wrapped. So, this is all about the protocol what you have to follow, and I would like to take you to my lab where the students are actually going to show you how to use the cytokine array to measure the different types of cytokines, and with this demo they will actually going to tell you each and every precautions what you have to take and what are the different steps what you have to follow.
Today I am going to demonstrate cytokine array is nothing but a protein array where you can detect multiple unlights from sampling. The sample source can be from cell culture supernatant or tissue homogenized sample or cell lysates. We can use serum or blood also. So it is generally the principle behind this cytokine arrays. Carefully selected capture antibodies spotted on a array membrane. When these capture antibodies comes in contact with the unlight, they will attach. Then again you have to provide detection antibodies, which will form a complex with your unlight, which is simultaneously detected with the chemiluminescence substrate. So today we are going to use R&D systems mouse cytokine profiler array kit panel A. So let's start. So I'm going to open this cytokine panel. 4 cytokine array panels. As you can see, there are blue dots. Each blue dot indicates capture antibody. So what we have to do, first we have to block these membranes. So in order to do that, we use array buffer 6, dispense 2 ml of array buffer 6 into each well, into these 4 well panels. So next thing is, you have to place these arrays in this array buffer 6. So carefully take out the membrane outside from that pouch into this well. This number, what you can see, it should face upward side. So when I keep in array buffer 6, as you can see, the blue color gradually disappears. So the blue color is disappeared. What we have to do is, we have to cover this one and keep on a shaker for 1 hour at room temperature. While the array blocking is going on, we have to prepare our samples. These are the cell culture supernatants. So first we have to centrifuge to remove any debris, and out of that we have to take out 1 ml of sample and make it up to 1.5 ml using array buffer 4. Okay. So I already centrifuged. Now I am going to add array buffer 4 to samples. Once you added array buffer 4 to samples, next thing you have to prepare detection antibody cocktail by adding 100 microliter of distilled water to detection antibody. So this I have prepared by adding 100 microliter. You have to suspend properly. So out of this you have to take out 15 microliter and add each sample. So let's do this. Here Now we'll keep this samples on a rocking platform with constant shaking for 1 hour at room temperature. After blocking the array membrane, we have to remove the blocking buffer. Next, we have the detection antibody incubated with the samples. Now we have to introduce these samples into membranes. So I am going to pour all this into this membrane. For every sample we have to use separate tip so that we will not get any cross signaling. Once you introduce a sample, carefully close the lid and keep it at 4 degrees Celsius on a constant shaking for overnight. So after that we have to incubate with the streptavidin HRP conjugated antibody, then we will develop. But that will be later on. Now we'll keep this rocker for overnight at 40 degrees Celsius. So we have incubated overnight with the sample, and now we have to remove solution from glass. After removing sample solution, we have to add wash buffer to remove unbound antibodies. Cover the lid and you have to keep 10 minutes on a rocking platform shaker. After washing, carefully remove the washing buffer from plate. Once you removed all the washing buffer, you have to add streptavidin HRP solution, diluted solution to this tray. So what you have to do, you have to prepare diluted streptavidin HRP as per the manufacturer instructions. This is the streptavidin HRP substrate. As you can see, dilute 1:2000. So I already prepared diluted solution. This is streptavidin HRP diluted solution. So out of this 2 ml you have to add to each array. So I am going to add this one. So during this process, don't let the blood dry. Once you added this streptavidin HRP, keep these plates on a rocking platform for 30 minutes.
Start.
So after incubation with the streptavidin HRP, now we have to wash at least 3 times using wash paper, then we will develop the blood. So these are washed and ready to develop. So we will use the manufacturer provided chemi reagent. So we have to mix chemi reagent 1 with chemi reagent 2 in 1:1 ratio. Then we will add it to array, then we will see it in chemi dark. So you have to add chemi reagent on top of it. Allow it to roll R over the blood. Then you gently swipe to remove excess chemi reagent. Just to develop this one. So while exposing, you have to keep constant for every sample to prevent any signal mismatch. So these are the signal exposure only. We are going to use normalization of the cytokine pixel density. So as we can see here, this is the actual cytokine. Whatever the red dots are showing, those are actually saturated pixels. So we can remove that one simply. So this is the cytokine array. As we can see, each dot represents one cytokine. On corners one, this is the this corners one. These 3 represents control dots, which we can use for normalization. And here 2 dots actually we cannot see, that is actually PBS1. So this is the cytokine array. During this process, sometimes we may not get signal, or we may get excess signal. So in that case, you have to look carefully into your protocol and all the buffers, whether you prepared right concentration of buffer, and also always use gloves.
So with this demo, I am sure you might have understood the protocol, what you have to follow, and how to detect the cytokines into the using the cytokine array. The results what you see after you are doing with this assay is the number of spots what you are going to see on the membrane. So you can imagine that you have 2 samples, one is untreated sample, the other one is a treated sample, and what you are going to see is these spots what is there onto the corner, and these spots are actually the reference spots. So what you have is you have a cytokine array sheet where they have given you that what is mean by the A1, A2 and all that. So what you see is that you have the numbering onto the left side, which is like A, B, C, D like that, and you have the numbers onto this side. So all these numbers are actually corresponding to the individual cytokines. For example, the A3 is A3 is corresponding to something, then similarly the A4 is corresponding to something. So, you know which spot is corresponding to what, and these are the reference spots. So, these reference spots are being used to align the membrane with the reference sheet, so that you will know which spot is actually corresponding to what, and reference spots are also being used for equalizing purposes. For example, if you are trying to compare this and versus this, then all the intensities has to be compared with each other using this as equalizing factors. So, how to analyze these results? So, what you are going to see is you are going to see a different types of spots, and what you have to do is so for example, these two spots are a part of the pair, like the two cytokines. Once it is a single cytokine in duplicates. So, you have to measure the intensity of this particular spot, and similarly you have to go spot into the treated sample also, and then you have to measure, and then by using these reference points you have to calculate what will be the fold of multiplications I have to use to equalize this spot with this spot, and then only you can be able to plot the intensity of this particular cytokine, whether it is upregulating or downregulating. So, positive signal what you see on the developed film can be identified the placing on the transparent overlay template onto the array image and aligning it with the pair of reference spot in the three corner of each array. Reference spots are included to align the transparency and to demonstrate that the array has been incubated with the streptavidin HRP during the assay procedure. So, this actually reference spots are being done simply because they are actually going to be a positive control that nothing is wrong with the streptavidin HRP system or detection system, and you are actually getting the reactions work. But for example, in some cases if you do not get the results, then you will not then the reference point also definitely should give you the signal irrespective of whether the sample is giving you a signal or not, because the reference point does not have the primary antibodies, they are simply having the other antibodies, and that is why the streptavidin will definitely go and bind to these points. Then you have to measure the pixel intensity on each spot, and then you have to use any image analysis software to compare the pixel intensity of the untreated sample versus the treated sample. You have to use these reference spot as the equalizing factors to multiply or to divide the signals, and that is how you can be able to compare to know whether which signal is over expressing or which signal is down expressing. For example, if I show you that this image, that what you see is that this signal is actually not present in this particular untreated sample, but it is very intense in the treated sample. Similarly, this signal is faint compared to this signal is very high, and what you see is all other these encircled signal, all these signals are pretty high compared to that they are very weak in the treated sample, in the untreated sample, which means these are the cytokines which are actually increasing when you are treating the cells with a particular with a treatment. This means these cytokines probably could have some role in driving the biological processes, or they might have role in driving the immunological responses. So, this is all about the different types of immunological tools what we have discussed based on the antigen and antibody interactions. We have also discussed some of the immunoassays what you can also use to monitor the immunological reactions, or you can also use them to answer some of the biological questions. So, with this I would like to conclude my lecture here. In a subsequent lecture we are actually going to discuss some more aspect related to experimental biotechnology. Thank you.